Okay, this is podcast number 3 for chapter 9 on the nervous system. In this podcast, we're going to take a look at how one nerve cell will talk to another nerve cell. So every nerve pathway, the impulse is going to travel in only one direction from one neuron to another, or from a neuron to an effector cell, like a muscle or something that is going to make an event happen. So nerve transmission, I said, always goes in one direction. So it will start at a dendrite, which would be like right here, and then it will move through the cell body, down the axon itself, till you get to the axon terminal. Then the impulse will come to the synapse, where a neurotransmitter, a chemical reaction, is going to occur, and then the dendrite of the next cell will be stimulated, and then we will continue on with this process. So what we're going to take a look at today is what actually happens in this area here, which is the synapse. So what's going on at that synapse is a pretty complicated series of events that are going to occur there. So we're going to take a closer look at that. So the synapse is the junction between two communicating neurons. So where one neuron comes together with another neuron, we call that spot a synapse. So basically it's the axon terminal of one neuron meeting up with the dendrite of the other. So here's your axon terminal meeting up with the dendrite of the next area, the next cell. Or it could be with just a piece of the cell if it was a muscle or a gland. Now the interesting thing here is that at the synapse, the two cells do not actually touch. There's a space between them, and special chemicals called neurotransmitters are going to be released into that space at the synapse, and they're actually going to do the communicating. So this is a way of sending information from one cell to another cell. So these special chemicals are actually going to send the information. So if you think about it, if you were passing a note to somebody, you would hand them the note, you wouldn't necessarily speak directly to them. So the synapse is working in the same way. The neurotransmitters are like passing along a note instead of saying something directly. So if we take a look at the structure of a synapse, here we've got the axon terminal of one cell, so that's the very end of an axon. And that axon is filled with vesicles that have neurotransmitters in them. So the little triangle things here are the neurotransmitters in this case. So the impulse comes down this axon, and it's going to stimulate the release of these neurotransmitters into the synaptic gap. This is also called the synaptic cleft. You'll see it both ways, but it's basically that space that I just said exists between two cells. So they're not actually touching. The neurotransmitters get released, they bind to receptors on the next cell. In this case we have a dendrite, so that they will bind to these receptors. This is going to cause special protein channels to open, and that's going to let positively charged ions like sodium rush into the cell, and the sodium is actually what's going to be used to send the impulse. So we can break this down into steps of what happens at the synapse. So in step 1, an impulse is going to travel down the axon of a neuron. So we've got that neuron that was there. The axon is the very end of it, so the impulse travels down. The vesicles that have the neurotransmitters move over to the cell membrane, and they release those neurotransmitters into the synaptic cleft or gap. The neurotransmitters will then bind to receptors on the next cell, so they will find their appropriate receptors on the next cell, and they will stick on there. Once enough neurotransmitters have bound, we reach something called threshold. At that point, the sodium channels will open. That's going to let the positive ions in, and this causes a new impulse to start. So in the next podcast, we'll take a closer look at the impulse. This happens in an all or none response. Once enough channels open and the impulse starts, the entire cell is going to go through the impulse. It doesn't just go a little bit. It doesn't start and then stop. Once an impulse starts, it goes all of the way. And that happens once you reach the threshold of the neurotransmitters. The neurotransmitters are then going to be broken down by enzymes, or they get taken back up into the sending cell. You don't want to let the neurotransmitters just hang out in the synapse, because they'll just keep stimulating the next cell, and you don't necessarily want your cells keep stimulating over and over and over again. So we're going to take a look at an animation of this process. All right, so here we have one cell that's going to be communicating with another cell. So remember, the impulse is going to travel down and then at the synapse they're going to come in contact with each other. So this is a closer look at the synapse. Basically I'm going to go through the steps that happen here. So here is this little star represents the impulse coming down. So the impulse travels down the end of the axon of one cell, and that stimulates the release of these neurotransmitters, which are the green dots in this case. They empty into the synaptic cleft, that opening in there, and then they will come over, they will bind to receptors on these special little channels. Once they bind, that allows the channels to open, and sodium ions can rush in, and that causes the impulse to stimulate the next cell. Now once that impulse is stimulated in the next cell, the neurotransmitters get taken up and they go away, and then the entire process can start all over again. Okay, so you can kind of watch it all in process. Now this takes a fraction of a second to occur, because remember how quick your reflexes are, so this happens very, very quickly. The neurotransmitters always have to go away, and then the cell is reset. Okay, so there are two types of neurotransmitters. Excitatories are going to be increasing membrane permeability to the sodium, which is going to basically increase the chances of an impulse being sent. So these kind of make your nervous system work a little faster. Acetylcholine is going to stimulate muscles. The monoamines like norepinephrine and dopamine give you a sense of feeling good. If you have low levels of those, that indicates depression. Dopamine is the main neurotransmitter that is stimulated by drug use, like in cocaine. Those types of drugs that give you a huge rush. Dopamine is the main one that is going to be stimulated in that case. And then there are also inhibitory neurotransmitters. These are going to actually decrease membrane permeability. So they decrease the chances of an impulse being sent from one place to another. Endorphins are an example. They reduce pain by inhibiting the receptors. So endorphins sometimes will be released. They make you feel good, but they often do that by reducing the amount of pain that you feel. Serotonin is another one that will kind of calm down your nervous system so that you get sleepy at night. So serotonin levels help with your sleepiness overall. But all the different neurotransmitters, their job is to help to stimulate the next cell in some way in order to keep your nervous system transmitting messages and controlling all the different functions therein.